There you go. Okay, perfect. Okay, hi everyone, I'm Minu. So I'm very excited to be here to talk about my baby. That is the blood vessel which I developed outside the human body. And thank you so much for Women Techmakers for having me here. I'm so honored to be here. So before I start talking about my baby, I would like to talk about a little bit about where I come from and my mom and dad. So this is where I come from. I don't know how many of you have been here. It's southernmost part of India. And yeah, so this is like this tiny little temple city where we have it's a very small suburb around this little hill. That's where I come from. So that's me and my sister. And I come from a very conservative society, but from a very progressive family. So that's my dad. He's my hoarder, I would say, because whenever the society just told him, okay, she's a girl, she's not supposed to do this, she's a girl, she's not supposed to do that, he held the doors so that their hands will not touch me. And he said, she's my daughter and she can do whatever she wants to do. So he's my rock. And this is my mom. She got married when she was 18 and she had me when she was 18. And she was a housewife until she was 40 years old. Then she decided to go get a college education. And now, after 15 years, she's a district tax officer for the government. So she showed me that even though you are from a conservative society, it is okay and it is possible to not only be a victim, but also can be a survivor and a winner. So this is when I got an award as a role model scientist from our research consortium. And the previous speaker was talking about how it is important to take small steps each day. And whenever I took a small step to reach this place, I got pushed back 10 steps. And it took me 1,000,000 steps to go from this to here. Then I came to Vienna. And after I came here, there were nobody to push me back, but I had my friends and people who held my hands, and even today are holding my hands and helping me move forward. And Women of Vienna is one of such a huge impact in my life, who are one of the important reasons for me being here. So I think it's important for us to be empathetic to all the people around us, and not only focus on our own growth, but also the growth of others. Let's all grow together. So that's my life so far. This is my professional journey. So I did my master's in biomedical sciences. Then I was working in studying proteins. Then I came to Vienna and I was a neuroscience research fellow for 2 years. Then I realized my heart is longing for something else. I want to look and be with living beings. So I decided to do a PhD in vascular biology, especially in tissue engineering. So every day, thousands of surgeries take place to repair or to restore damaged organs, which is caused due to disease or trauma, etc. So imagine how many donor organs we need. So what we do, we tissue engineers, we try to grow those tissues and organs in the lab. How do we do that? We harvest human cells from the patient, and we use scaffolds like 3D printed scaffolds or bioengineered scaffolds, or even human tissue scaffolds like decellularized tissue scaffolds. And we add many biological factors and use all these three together and create kind of a human body-like environment to develop these tissues, which then can be used in clinical transplantations. That's the priority. You can also grow meat, which is actually happening. So soon we will have lab-grown meat in your hamburgers. And so you can have a lab-grown leather. You can have your own organs. Whenever I develop my own blood vessel in the culture room, I always name it, I talk to it, it's like my own pet, and they talk to me back, but that's a So you can have your own organs on chip, which you can use to understand what these guys, believe me, these guys talk to each other. The cells in your organs, they are having fights, they are having romance, they are having everything. So so you can have organs on chips, and I'm not quite sure about what this is, but because there is robotics there. So how are these models made? The main thing is, so basic cell culture. How do you grow these cells? You harvest these cells from the body, and you seed them on a two-dimensional plate, right? But this is not enough to develop an organoid, because the cells in your human body is in a three-dimensional environment. So we have to make 3D models so that the cells receive the growth factors from all the sites in three-dimensional in a three-dimensional environment, and also the cell-to-cell contact is not only limited here but all over. So this is a challenge to make the cell grow in a three-dimensional environment. So far, people have not really succeeded in making a 3D model of a vessel wall, which is really lifelike, until Minu came along. So basically, why did we do that? Because science is expensive. My boss is not going to tell me, hey, make this. No, there is a reason why we did that. So, you know, when we have coronary artery disease, and then you have a plaque built up in your blood vessel wall, what happens is your blood is not going there because the lumen size is completely reduced. So how doctors treat this is they bypass a graft from different part of your body. How awesome is this, right? They cut this, this saphenous vein is from the leg, so they bypass the blood flow so that the blood doesn't flow through the blocked artery but through the bypassed artery, right? This is awesome. And then you go home, everything is fine, and after 5 years, you get into trouble again because, so the human body doesn't know what we did, right? What it thinks is that this is a wound according to the human body, because come on, this is not supposed to be here. It's supposed to be in the leg. You can't have it in your heart. So the human body thinks, oh my God, this is not right, so I'm going to cure it. So what it does is like it treats it as a wound and starts building up cells. So when you have a cut on your hand and you have platelets, you know, the cells which prevent blood clotting, and so it starts making a scar tissue. So it makes a scar tissue here in this vessel. So your own body is trying to kill you. So it's like, okay, I'm going to save this person. This is not right. So what our research was that my boss told me, okay, we need to make sure the cells here don't overact, and we need to calm them down. But how are we going to do? I'm not going to have human patients on whom I can experiment, right? So what we usually use is rats and mice. And still, it's not very humane to use. It's not so easy. It's not like we have so much rats and mice on whom we can just play around. It's a lot of money and a lot of ethical issues. So my boss wanted me to develop a blood vessel in the lab. So what do we need for that? So this is a structure of a blood vessel. Okay, so this is the longitudinal section. So this is actually like a hole, and the blood goes through the tube here. So this is a layer of cells immediately. These cells are called endothelial cells, okay? These cells are very important because they are packed so tight so that the blood doesn't get into the tissue. So sometimes you get these like internal hemorrhage or vascular leak. These things happen because these guys move away from each other. So these are the most important cells. Much more important to them is these cells next. These are my favorite cells. These are called the smooth muscle cells. They are so beautiful, like beautiful. And these guys are important because they are involved in the contraction of and the relaxation of the blood vessel. So they contract and relax the vessel wall. So these guys prevent unwanted leakage into the tissue, and these guys are there to give the structural integrity and function and aid in the function of the blood vessel wall. So our model, so these are how the cells look like under the microscope. So who do you think is pretty? I think they are prettier. I don't like these guys so much. So you can clearly see that they are tightly packed. So these guys form this layer, and these girls form this layer here. So can you guess where did I get these cells from? Any guess? Anybody? From human, yeah. But where?
Okay.
No. So this is a umbilical cord. So we get fresh. I reduced the gore as much as possible. Like, there's not so much blood in there, but it's so bloody when I work with it. So really. And then we go have lunch afterwards immediately. So we get a fresh umbilical cord. It has to be fresh. And what we do is we cut it open. It's not easy. You need to, it's really tight, right? And you have to fight for it and really remove it. And then you see the arteries which go to the baby here. So you have the vein here and you have the arteries here. So we remove these veins and arteries from the jelly, and then we digest the cells which are inside these blood vessels using enzymes called collagenase, or these cells inside the body they are connected together with the help of extracellular matrix, so called like collagen and all these. So it's like a spider web. You have the spiders all over, and then you have the web. So that's important. So how we disrupt the cells is by using these enzymes which eat away the extracellular matrix, and then the cells come out. Yeah, and this is how they look on the first day, and then they start growing. I said they grow, they dance, they talk. Sometimes they get contaminated and they ruin my day, but that's all in the game. So these are the smooth muscle cells actually. So we have the cells now, but we have a problem, because that's the fun about science. So the problem is that I told you the cells, the smooth muscle cells, and those look like these, right? Very thin and long and beautiful. But they don't actually look like this all the time, because they have two different characteristics. They become angry and they start proliferating and acting up, or they become quiet and calm and beautiful. So we need them and we don't need them. So my important thing I need to do is that I need to calm down the smooth muscle cells. That's why I sing to them sometimes, but I'm a very terrible singer. But sometimes it works, so that they don't get activated and proliferate and start proliferating, because if they do, then they do not let the other cells we talked about, these little tiny cobblestone-like cells on the top, they don't let them grow if they are activated. So I did a lot of science here to calm these guys down. And here you have these guys here. Now they are calm. What I do is I take a Petri dish, I coat the dish with collagen, I seed the cells, smooth muscle cells, which I isolated from the umbilical cord. And after 4 days, they are like this. They are like growing all over the place because they are happy and they want to eat all the food and be all like a teenager. Then I starve them. I don't give them any food. I starve them. After I starve them, they are like, okay, I will behave now. And then they calm down. They calm down and they become beautiful like this. After 5th day, I take this other little tiny cells which I don't like, and I seed them on them. And by day 7, those tiny cells have formed a tight monolayer on top of this multiple layers of the smooth muscle cells. So now I have a 3D vessel wall, which has both important layers of a vessel wall which is inside our human body. So this is how it looks like. Sorry, is it too loud?
It's in the recording.
Okay, so this is how it looks like in 3D. So to have this picture, I spent 3 months. So what we do is we stain the cells, to say it simply, with different dyes. So these are the, can I ask you, do I like these cells or these cells? Which is my favorite, the red or green? No, the red. The green is on the top, those tiny ones which are so tight with each other. Those are the guys who are on the top, and the red below, those are the smooth muscle cells, which are these long, tough to calm down cells. They are my favorite. So you can see here. So we have the monolayer. So if I wrap this up, right, this is like in a plane. But if I wrap it up, it will end up being a tube with these cells inside, which kind of like this. Yeah. So these are the green cells now, and these are the red cells now. Yeah. So do you get it? Yeah? Okay. This is my first time when I'm not talking to room full of biologists. That's why I'm like, so everything is clear? Yes. Okay. So next thing is, then it's a lot of validation, trying it, like adding stuff to it and seeing whether it behaves the same way like a real vessel wall, or removing stuff from it, and a lot of molecular biology, a lot of protein studies, and lots and lots. But this is something very interesting. Like I said, a cell needs extracellular matrix to survive. So it needs these connections. It's like women Techmakers connecting me to you all. So without them, I'm not going to stay here and talk to you. So what we found is that although these cells had only one extracellular matrix added to it artificially, that's collagen. But when we detected for more than 10 extracellular matrix proteins, we found them all in the model, which means the cells were living happily ever after. They are secreting stuff, they are having their own extracellular matrix, they are talking to each other, they are happy and healthy. So this here, you see the red cells are the smooth muscle cells, right? Yeah, is it right? Yeah, and the green are the endothelial cells. So these are the cells that protect us from vascular leakage, and these help in the contraction. And the white here, like the spider web, that's one of the extracellular matrix protein, which is found in the blood vessel wall, which is developed outside the human body. How cool is that? So, a little video. This is like a 3-dimensional rendering of the whole model. So basically, you can see the yellow is the extracellular matrix. I'm not sure if it's very visible. And the blue dots are the nuclei of the cells. It's not visible enough. Okay, the green, you can see the green cells on the top, right? And then you have the nuclei, and then the yellow is the extracellular matrix, and there are red cells behind, which I'm not sure if it's visible. Yeah. Oh, okay, because it's, yeah, okay, because it's really, really nice. I have spent like hours sitting in front of my microscope, looking at this and just smiling, like, oh, my baby, I'm so proud of you. Yeah, so I think the red is still not visible, but I think it's nicer now. Thank you. Yep, so you can fold it. If I seed it in a scaffold, in a 3D printed, a tube-like cylindrical scaffold, and I do the same thing there, then we have a tissue-engineered vessel wall, which we can then use instead of a biological graft. So this is how it looks like. So you have the green tight cells. These are the red cells. This is the extracellular matrix all over. And then you have the top cells. So all these are the characteristics which we needed to study to prove to the people that yes, it is real, it works like a real vessel wall, not just looks like, but also works like a real vessel wall. So it is not proliferating. It's not okay if all the cells in a vessel wall, so cells proliferate, cells divide. But in a vessel wall, the cells should not divide, because imagine if all, then it's called cancer, right? So we have to make sure that the cells are not proliferating. And also the morphology should be the same like how it is in the real life. It has to have a 3D distribution. It should have its own extracellular matrix, which is produced. And quiescence means like it should not react in a bad way to any kind of external environment, because that's how it is inside our body, that our blood vessels are just doing their job and being quiet and not acting up. So that's good for them. And then you have, but it should not also be a sleepy phase and not respond to anything else, because we also need our body to respond to stuff so that we are ready when some kind of a pathogen comes in and we are ready to attack. So we did a lot of tests and it responds wonderfully. And also we use this model to study how these two cells are interacting with each other. Like for example, if I add a pathogen on top of the green cells, the red cells react after like 3-4 hours, which means the green cells are communicating that, hey, there is danger on top of me, so let's get together and do something about it. So this talk got me like a lot of awards, but this is particularly, I'm quite proud of this because I represented Austria in a European postgraduate conference, and yeah, I won one of the best talk awards. This is from Hungary, and she's from, I think, Iceland or something. And I'm from Austria, can you see that? Yeah. So what we do is we isolate cells from the patients, we grow and differentiate them, we use scaffolds to engineer tissue engineering of blood vessels, and then we use those tissue engineered blood vessels in the graft surgery, because a patient doesn't have so many other blood vessels in his body. We can't just call a patient again and again, okay, we are gonna take this vein from your leg, okay, it fails, okay, this vein from your leg, okay, it fails, okay. Then maybe from your hand, it's not really nice to do that. So these guys come in handy, and a lot of tissue engineering, engineered by blood vessels are being used currently in cardiac surgeries. And this is my work, and I had such a pleasure talking to you, and I hope I made it interesting. Yeah? Okay, then it's good, thank you.